As concerned the genetic manipulation or a mutation, there are two choices. One is the random mutagenesis and other is the site-directed mutagenesis. In this module, we will talk about the site-directed mutagenesis. But before talking the site-directed mutagens, I want to describe here the somehow about random mutagenesis. Whenever we have to apply the random mutagenesis either with the physical agent or with the chemical agents, first we have to chalk out that range or that intensity of the physical agent in which that organism is fully killed. We have to make the survival curve or a kill curve. So if there is a kill rate or a percentage above 99% kill is achieved, or there is only 1% of the survivors, then there is a chance of genetic amendability. Then we can produce any mutant. So because when there is a harsh and there is an effective on a DNA, then there is a shuffling of that. So in random mutagenesis, we have to first chalk out the kill curve or a survival curve. When we have a survival curve or a kill curve, then accordingly we have to give a maximum doses and then maximum repeated mutations for getting a desired and positive mutant that only get and pass through our selection criteria. But as concerns the site-directed mutagenesis, many researchers seek to introduce the selected change to DNA in a site-specific manner. Analogues of nucleotides and other chemicals were first used to generate localized point mutation. So such chemicals include aminopurine, which induce AT to GC transitions, while the nitrosoguanidine, bisulfite and N4-hydroxycysteine may induce GC to AT transitions. So these techniques allow specific mutations to be engineered into a protein. However, they are not flexible with respect to the kind of mutants generated, nor they are as specific as later method of site-directed mutagenesis. But this mostly also come in a random mutagenesis, but to somehow we can call it as site-directed mutagenesis, but their effect is very random. So therefore they have some degree of randomness. So current technique for the site-specific mutation commonly involve the using of mutagenic oligonucleotide in a primer extension reaction with DNA polymerase reaction. So this method allows for point mutation or detection of insertion of small stretches of DNA to be introduced at specific sites. Advances in methodology have made such mutagenesis is now relatively simple and efficient process. Then is the protoplast fusion. So the protoplasts are formed from bacteria, fungi, yeast and actinomycetes when dividing cells are caused to lose their cell wall. Fusion from the mixed population of the protoplast is greatly enhanced by the use of polyethylene glycol. The method has great industrial potential and has experimentally been used to achieve a higher yield of antibiotics through the fusion with the protoplast from different fungi. Protoplast fusion has been demonstrated as an efficient way to induce heterokaryon formation and recombination with high frequency. Protoplast fusion has been successfully done with Bacillus subtilis and Bacillus megaterium among several species of Streptomyces species like S. coli color, S. acremimycini, and these are the different by which between the fungi, Geotrichum and Aspergillus and the yeast. Kim et al. 1998 did a comparative study on a strain improvement of Aspergillus oryzae for protease production by both mutation and protoplasm fusion. So he produced a mutant that have the 14 times higher yield with UV radiation, as concerned when you use ethyl methyl sulfonate that have a 39 times higher yield. But when he used protoplast fusion using polyethylene glycol and calcium chloride, 82 times higher yield of the protease he reported. You can see in this slide that how the protoplast fusion and the bacteria grown on plasmids are introduced into the animal cells via fusion with bacterial protoplast. So the bacterial cell wall are removed using lysozymes, and these are the different techniques by which the foreign material is entered into the new one. So the plastids are mostly be entered into the new one. So auxotrophy is the inability of an organism to synthesize a particular organic compound required for its growth. So an auxotroph is an organism that displays the characteristics. So the prototrophy is characterized by the ability to synthesize all the compound needed for the growth. So prototrophic organism is wild type organism expressing its natural phenotypes. So when we having a strain improvement strategy, then we produce different auxotroph and prototrophs. So then on the base of the auxotroph and prototroph, then we have a specific media to identify those mutants. Either they are auxotroph or they are a prototroph.